April. All right, so today we have a little unique. We have actually someone who's with us remotely. We have Damian Fermin, who I believe is a former graduate student here, and is now at Yale. Is that correct? At Yale. So he's going to talk to us about LuciPHOr 2. Take it away.
All right. So first off, I want to say thanks for having me. And my unfortunate feature of my talks is that I tend to talk fast to get to the end of it. So if you hear me going too fast, someone speak up, slow down, because I can't see you guys in this presentation. You have the dubious pleasure of having to see me, though. So anyway, as you mentioned, the title of my talk is going to be about Lucifer 2, which is a tool we made at U of M before I left. That's why I kept U of M here. And it's a tool for post-translational modifications assignment in mass spec data. I want to put a bit of a caveat here. I originally wrote a program, published a paper on something called Lucifer 1. And that was a program that specifically focused on phosphocyte localization. We wrote it in CPP and C++, and and I also wrote it to work with the transproteomic pipeline output, which is PEP-XML files. Out of that grew Lucifer 2, and the primary difference is that, we removed the restriction that it had to be phosphorylated data, and now it'll work with any PTM you can think of, and also the software is written in Java instead of TPP, so it can run on any operating system. And the last one is that you don't have to use the TPP; it'll take output from anybody. So although the talk is entitled Lucifer 2, the data I'm going to present is about Lucifer 1, because that's what we tested both versions on, and conveniently it illustrates our points. So without further ado, first a quick overview about what I hope to cover. So I don't know what the audience makeup is, if you guys are all RNA-seq people or not, or any proteomics people. So a bit about protein phosphorylation, mass spectrometry, and proteomics, and the bioinformatics of it. Then talking about the phosphoproteomics challenge, some previous work done in this field, and then what Lucifer brings to it.
So Damian, can you hear me?
Yeah.
It's Gil Oman. Welcome back, even from the distance. You have a very nice turnout here at faculty and students. So give us your best shot and tell us a couple sentences about your position. What are you actually doing there? Yeah.
Like now, or you want to towards the end, or?
Right now. Part of the introduction.
Okay. I currently work for Dr. Stephen Kleinstein in the pathology informatics program here at Yale. His research focuses on computational immunology. The projects that specifically I work with are on influenza immune response, and we're trying to we're looking at microarray data and flow cytometry data to try and decipher the characteristics that make some people unable to acquire immunity given the influenza vaccine. And that's the primary focus. Can we use big data to try and determine what characteristics these people have that prevent them from being immune to the virus? And if any, as a side project or a long-term project, perhaps we can develop or identify the characteristics that would make for a one-time flu vaccine instead of having to change it every year.
Yeah, great. Well, very good. Thanks for sharing that. Okay, please go ahead.
So as I mentioned earlier, this is a quick recap of why protein phosphorylation is important. The phosphate group gets attached to proteins, and when it does, it acts as a regulatory mechanism for many proteins. Some proteins become active or inactive based upon the presence or absence of a phosphate group in certain positions on its amino acid sequence. If you just take a quick look at UniProt, there's over 500,000 potential phosphorylation sites if you just look at protein sequences in humans. The database also reports 25,000 phosphorylation events on various proteins and their isoforms. One of the key proteins in this business is kinases. These are enzymes that add phosphate groups. And to give you an idea of the importance, there are estimated to be about 500 protein kinases encoded in the human genome, and they're a major target for pharmaceutical research. We know this because deregulation of kinases often lead to many diseases in humans, and for that reason, lots of cancer therapeutics are kinase inhibitors. I think something around 25 FDA-approved drugs are kinase inhibitors. So protein phosphorylation is important to understand and to study. So, and now I want to give a quick overview of mass spectrometry. I know this is a bioinformatics talk, so I'm going to try and go quick over this. The basic idea is you have some biological sample, you have to separate it out, and once you get your proteins, you digest them with trypsin, and the peptide fragments are injected into a mass spectrometer. The mass spectrometer first identifies peptides based upon their most intense peak, which corresponds to along the x-axis to their mass. This is the MS1 phase. And this peak is selected for further fragmentation, where we try and identify what the sequence is of that peak, and that's in the MS2 or MS stage. And now the bioinformatics starts in, so the mass spectrometer spits out this MS spectrum, but you don't know from the get-go, you don't know what peptide sequence this belongs to. And if you're really patient and really good, you can sit here and manually figure out the distances between the peaks, and then you can figure out what amino acid sequence corresponds to this. Fortunately, bioinformaticists have invented computer software, which are called protein search engines, which can match this spectrum to any candidate peptide sequence, so long as the peptide sequence is within the same mass tolerance of what the original peptide was. These search engines all work pretty much the same way. You give it a list of candidates, and they try to match it, and the peptide sequence with the best score is what it spits out. And we call the best match the peptide to spectrum match, or PSM. And that's the gist of the proteomics side, the bioinformatics side of proteomics. Now, there is an extra challenge when you're dealing with phosphoproteomics. So here again is the MS1 followed by the MS2 phase, where you fragment the peptide ions, and you get what is called the ion ladder. So if this is the peptide sequence, it gets fragmented along the back chain, and you get these peptide fragments. The ones on this side of the space are called the B ions. These are the Y ions. And if this was a phosphorylated peptide, when it entered the MS2, the phosphate group gets kicked off because it's pretty labile. And this produces what's called a neutral loss peak in the spectrum, which is represented here, this huge peak in this spectrum. The presence of this peak is so intense that often the other signal peaks which you use to identify the peptide become very low intensity relative to it, or become grass. And not only does it make it difficult to identify the sequence, but more importantly, you have a hard time identifying where the phosphate group originally was. So this is called the phosphosite localization problem, and most search engines have a hard time correctly localizing the phosphate group on these peptides. Here's an example of, this is the spectrum that goes with this peptide is doubly phosphorylated, and here you have the neutral loss peak. You can imagine it's pretty difficult to look at this and figure out the correct site for phosphorylation. And so, manually you could do this, but when you have thousands of spectra, that's a daunting task. So bioinformaticists have come to the rescue and come up with different ways of dealing with this. There are, as of right now, there's a ton of different ways of doing it, but these are probably the most popular and most effective in the literature. The phosphosite localization tools can be pretty much broken up into one of two categories: spectrum-based methods and search engine score-based methods. And I will talk more about these two categories in a moment. But these are the current tools in the literature prior to Lucifer. So I'm going to talk about, the first one is the spectrum-based methods, and I'm going to talk about A-score as an example, primarily because A-score was the first, and the other two methods in this category, PhosphorS and Andromeda, are really just different versions of the A-score algorithm and the A-score approach. So A-score came out of Gigi's group in Harvard, and the algorithm is pretty simple. So let's say you have this peptide sequence, and the red spots represent a phosphoserine in this permutation, a phosphothreonine in this permutation. And you want to figure out, well, which of these is it? So if you have the spectrum, what A-score does is it breaks the spectrum up into bins of 100 m/z windows. And within each bin, it takes the top n most intense peaks within each window and tries to match them to the phosphopermutations. So if you can imagine, here's the spectrum, you break it up, and you overlay the B and Y ions and try and match them. And each permutation is scored using a binomial probability, a cumulative binomial probability. And you do this iteratively, increasing the number of peaks you allow within each bin. And eventually you hit a peak in the peak depth, meaning you've come to a number of peaks within the window that's optimal and gives you a maximum score. The algorithm then chooses this peak depth and then computes the score, scores the peptide, this peptide permutation at this depth, and compares it to the other, the next best match. So in this example they present, the depth was found to be about 6. So they score they try and match all the peptides, fragment ions for this permutation. They're able to find 5, and in the next best permutation, which is this one down here, they were not able to match any of them. So the final score is the difference between the two of them. And like I said, phosphorescent and Andromeda do pretty much the same thing. They just have different, either they use different windows or they use a different value as their final score. The second method is search engine scores, and there's only really 2 of them: the MD score, or the Mascot Delta score from Kuster's group, and the SLIP score, where the SLIP score is site localization in peptides. And these methods directly use the database results that you get and use these to interpret use these to come up with a localization score. So how they work is they simply take the difference between the top 2 permutations for a peptide, and the difference between the two is the delta score, and this is considered to be the confidence in the assignment. So the larger the delta score, the more confident you are in the assignment made in the top score. So if you have these 3 possible permutations for the sequence, and this site is the correct one, let's say the search engine says this is the best score, the next best score is this one, so the delta score is 5. And so you would say that's how confident you are in the assignment made in this case, in this permutation. So what I did is I took the best of both worlds. I tried to take this, I borrowed from the spectrum world of A score and the Mascot Delta score, and I published a program we call Lucifer here. So Lucifer provides phosphosite assignments, but it also provides a false localization rate for the assignments it makes. Here is a layman's overview of how Lucifer works. I'll go more into the details in a moment, but this gives you an overview in case you find the next slide a little more difficult. So here you have, let's say, the search engine output. This is PepXML files, and these are the actual spectrum files. And these are the 2 things that Lucifer needs to work. So once you have both of these, you feed them into the program, and you first filter out PSN matches, peptide to spectrum matches, that you don't want to score because they're just low scores or bad quality. There's no point in scoring bad data. You're not going to use it anyway. Once you've done that, the next step is Lucifer selects the high scoring PSMs and for model parameter acquisition. And it does this in a charge dependent manner. Once it has these parameters acquired, it goes back and scores all of the PSMs using these parameters. All right, that's the gist of how the program works. The parameters it acquires are the intensity of the peaks and the M/Z accuracy of the matched peaks. All right. So this is the more detailed approach. So as I mentioned, the first thing we do is we learn the features of the data from these distributions. So one of them is log. One of them is the intensity of the peaks, both the matched peaks, which are in red, and the random peaks, or the unmatched peaks, which are in blue here. In addition, we compute the mass accuracy of the matched and unmatched peaks. So here again, red is the matched peaks, and blue is the random peaks. With this information, we go through and score each phosphopeptide permutation, and we assign it a raw score. So here's our example again. This is the peptide sequence, and we take these are the raw scores assigned to each of them. So let's assume this is the spectrum we got for this peptide sequence. What we do is we identify the peaks that we can match for the top 2 permutations. So in blue, I don't know if you can see that it's blue, but these peaks are the matched peaks for the blue permutation with the phosphothreonine. And the red peaks, which I doubt you can see, here's one, here's the other one, are the peaks that are unique to the phosphoserine permutation. What Lucifer does is it matches all the peaks it can. Don't worry if you can't see this. I'll show more closely in the next slide. Matches all the peaks it can for each permutation, sums up their scores, and then their difference between the top two permutations is called a delta score. And we use this delta score to estimate the false localization rate. So, oops. So here's that table up close again. Here's the blue peaks and the red peaks. And what you see here in bold are the peaks that are unique to this permutation. And you can see here is the intensity for those peaks and their distance. And by distance, I mean the mass M/Z accuracy, which is the distance, the difference between the observed M/Z value for this ion and its theoretical M/Z value. These two values are used to compute a log odds score, which is reported here as the ion score. And the cumulative sum of all these match values is the final raw score for this permutation. The same thing is done for the other permutation, the phosphoserine. And if you don't see a match peak value here, it's because you didn't match it or it doesn't belong to this permutation. And the same thing is performed. In the end, you take the difference, and that's called the Lucifer delta score. So once we have the delta score, the software has also made an assignment of the phosphorylation site. The problem is that this output is still only a prediction. Okay, there's no guarantee that the site that Lucifer reports in the top scoring permutation is correct. And this is true for all of the tools. So we had to come up with a way of estimating the confidence we can have in the site assignments that Lucifer made. And for this, we use a false localization rate, which is measurement of the percentage of incorrect matches or incorrect localizations at a given score threshold. So FLR is nothing new. It's a pretty common feature in proteomics. Usually it's called false discovery rate in that context, it's used for trying to determine how accurate the assignment between the particular peptide sequence and the spectrum it's assigned to. So we've just extended that same idea to the phosphosite localization. Different tools try to do this in different ways. Andromeda, for example, which is like a spectrum-based method similar to A-score, it just accepts the FDR that's used for peptide sequences and interprets that to be the error rate for the localization. The slip score by Chalky's group is a little more interesting. They allowed not only phosphorylation of serine, threonine, and tyrosine, they also allowed for the protein search engine to phosphorylate proline and glutamic acid. These are two residues that don't normally undergo phosphorylation. And so whenever they observed these cases, they knew it was a bogus hit and they could use it to estimate the false localization rate. None of these two methods are particularly good. The Andromeda method primarily because if it works for decoy peptides, that doesn't mean it's the same model for site localization. The slip score, although it works, the problem is you have to incorporate these parameters into the initial search. And you can ask any mass spectrometer or proteomics person, the more amino acids you say are modified, the longer the search takes, and nobody wants to wait for data. So Lucifer takes a different approach. So for a given peptide that has K STY modifications reported in it, the Lucifer will generate a new peptide sequence with K non-STY modifications, which are decoy matches. And we score these decoy matches using the same algorithm as if they were real ones. All right?
Well, explain that. Explain what is a non-STY? It means it has to be a P or an E or something else?
It can be any amino acid that isn't already modified by some other modification, like oxidation or carboxymethylation. As long as it doesn't have some other modification, it's a candidate to be artificially or synthetically phosphorylated for the purposes of estimating the false localization rate. Maybe this example will help. So I'm going to show you two PSN matches, and they're for the same peptide sequence, but you have to envision the case 1 is for one PSM and case 2 is for a different PSM. They just happen to belong to the same amino acid sequence. And so here we have the 3 possible valid phosphorylation events for this sequence. This S, this T, this S, this T, okay? The decoy permutations are all the other residues in the sequence. So what Lucifer does is it first scores these guys, and immediately following that, it puts the phosphate group on this residue and tries to score the theoretical product. And it does it for each of these and records their scores. Okay. Then it still goes back and scores only the top 2, computes the delta score from the top 2 hits, which in this case happen to be these 2 cases. The raw score for the top hit and the second best hit. Now in case 2, this is again, it's going to be the same sequence, but it's a different spectrum that was matched. In this case, again, the top scoring the real permutations are up here, and the decoys are below. But you notice that the raw scores are overall lower, which means probably fewer good peaks were matched. But regardless, in this iteration, the top scoring permutation is a decoy, right? If you're phosphorylating, if you're using phosphorylation, because valine does not undergo phosphorylation, at least not naturally. And so what Lucifer does is it takes this as the top score and then looks for the next highest value, which is this permutation. So the delta score becomes 1.74. Does that help, Dr. Oman?
No, I'm really confused now because it's not just that it shouldn't happen, it's that there's no chemical site for phosphorylation of valine and leucine. So how do you get confusion about the spectrum that would make that decoy seem plausible?
Well, you have to remember that apart from the true peaks that represent fragment ions in a spectrum, you also have noise peaks, which are not valid or just are present by chance.
Right.
So those peaks are still eligible to be matched, be they to real fragment ions for the real permutations or for any of the decoy permutations.
Okay, and the delta score here on the right, case 2, I would have expected would be minus 1.74 because you're comparing the decoys against that.
Well, it's the difference between 3.38 and 164. Because it's always, the algorithm always takes the difference.
It's the absolute difference, not the directional.
Right. Comparison. So it always takes the difference between the top hit and the next best hit. It's always a positive value. Does that help?
Other questions? Other questions?
You have to speak up because I can't see you if you raise your hand.
Okay. For your case 1, so, I noticed that there were 2 target permutations that are actually above your decoy permutation. Is it possible that both of them are correct? Or like, let's say you have 2 phosphorylated sites and both of them co-elude into your second phase of the mass spectrometry?
Well, usually based upon the mass of the peptide, you know how many phosphorylation events are present. So I'm showing a simplistic example in which we know from the beginning, from the mass spec output, that the peptide only contains one modification. So we only, given that from the beginning, then we only consider the possible condition in which there is only one modification in each of the possible permutations. If there were more phosphorylation events, then there would be more permutations, obviously, or fewer in this case. Does that help?
I guess I'm asking a different one. So let's say, is it possible a single peptide contain 2 potential phosphorylate sites and both of them get co-eluded into your second phase of mass spectrometry?
Yes, that's a common biological event. So you do get peptides that are phosphorylated. Both of them get stuck because they have the same mass, so they both get selected and they both come up in the spectrum.
Yeah. So
And
in
so
this case, yeah, in this case, I noticed that, for example, like your highest score in decoy permutation here is like a 68, while two of your target permutations have higher scores there. So is it possible that the actual spectra is a combination of two different kind of phosphorylation here?
yes, that is possible. But and when that does happen in practice. I haven't demonstrated that here just to keep things simple, but you do get examples where two possible peptides end up in the spectrum, and it's very confusing and very difficult. But the general result is the delta scores are very small because you have peaks that can match to both cases, and so it's an ambiguous situation, and you can't tell them apart.
Thanks a lot.
So once we have the decoy information, we use it to construct an FLR. So the first thing is we plot out the distribution of delta scores for both the non-decoys or the real data and the decoy cases. The decoys, we expect them to be few and infrequent. And if they're not, better go back and look at your data, something's wrong. And once we have that, we fit a mixture model to both distributions. From this, we then pick a score threshold, and the false localization rate is the proportion in the third panel of the decoys at that threshold to the number of non-decoys at that threshold. And that's how we estimate the FLR. So all of this is very nice, but you actually got to put it in practice and try it out. So fortunately, Kuster's lab in Belgium was very generous to provide us with a synthetic library of known phosphorylation sites. They published a paper on it in which they described the Mascot delta score method back in 2011. And they were kind enough to provide us the raw data they used along with the correct phosphorylation events in their synthetic library. This library was composed of 180 synthetic peptides in which we knew where the site of phosphorylation should have occurred. The library can be further broken down into 164 singly phosphorylated events and 16 doubly phosphorylated events. At the time, the field had a mixture of both high mass accuracy data, which is CID, and HCD, and then low mass accuracy data, which is called CID. I think I could be wrong. I've been out of it for a while, but I think that the industry is going primarily HCD. But at the time, the data, we had low mass accuracy and high mass accuracy mass specs, and I'm pretty sure people still use both. So fortunately, the library was running both ways, was analyzed using both types of mass spectrometers. So we got the data, we searched it using our search tools, and then we compared Lucifer's performance to two other methods, both A-score and the Mascot delta score, represented here as MD score. So along the y-axis, you have the fraction of sites that we identified correctly, and then the x-axis is the false localization rate. The numbers in parentheses tell you how many PSMs were matched by each of the search tools. So in the CID data, the low mass accuracy, at the critical FLR of 0.01, Lucifer, came out ahead. And in the HCD, we also came out ahead, although, as the FLR goes out, we don't do as, Mascot delta score outperforms us. But, we like to stick to the 0.01 I'm conservative with my estimate, so 0.01 is the critical cutoff that we use. Because we knew the true phosphorylation sites, we could look at our estimate for the FLR versus the true FLR with this data set, and we see that we're doing quite well for the CID FLR accuracy. And we had a bit of a hiccup here in the HCD, and it looked like we were underreporting the FLR. So this was a bit of a problem, and on further examination, we thought that the big issue here was simply the size of the database we're using, the size of the data we're using. we're only talking about 1,000 PSMs, which any mass spectrometer will tell you isn't a lot to work with. And it's only 180 peptides. So we couldn't really go any further until we found a larger data set. And Kuster came to our rescue yet again. Back in 2013, he published another paper in which he made a much larger high mass accuracy HCD library. And again, he was kind enough to provide it to us. This library consisted of 57,000 phosphopeptides, and they were all singly phosphorylated, and they were all done in high mass accuracy, which was the technology that we were having issues with. So once we had gotten the data, we ran Lucifer through it again and compared it to the Mascot delta score. And here you can see we did a lot better. We did better than Mascot delta score again, but more importantly for us, we corrected the underestimation of the FLR. So that made us feel good, and we were able to continue with software development to lead to publication. So in conclusion, I guess Lucifer implements a novel scoring algorithm based on probability model using the peak intensities and the mass accuracy of the M over Z. And unlike many of the other methods out there, Lucifer provides a false localization estimate at the peptide level for the PTM assignments it makes. And like I said, Lucifer 1 was originally released only to work with primarily with the TPP. So you had to use TPP and then you could use Lucifer. Lucifer 2, though, has had that restriction removed. It can now, as long as you can get your data in the right format, which is pretty easy, it's just tab delimited, you can use it. And also, it's now written in Java, so you can just download the jar and run it. So I would like to, first off, I'd like to thank you for your attention, and I would like to thank Dr. Bernard Kuster, who, provided us with those 2 synthetic libraries, and without them, we wouldn't have been able to test our algorithm and verify the results we were getting. At U of M, of course, I want to thank Dr. Nesvizhskii, who was my boss when I was working on this project, and Dmitry Anatolov. He was a great Java programmer who helped us find a lot of bugs and speed up the program immensely. Dr. Hyung Hwang Choi over at the National University of Singapore was a statistician who helped, a friend of mine in this project. He was the one who developed the FDR model that was used to estimate the FLR in the program. And of course, I would like to thank Alex, because without him, you wouldn't be seeing this presentation and my lovely face. So this work was supported with money from the NIH and from the Singapore Institute of Science. So if you want to download the software, you can find it at that website. That's for the latest version, Lucifer 2. You can still get Lucifer 1 as well if you want that. So if there's any questions, that's it. Thank you for your attention.
Questions?
Let's see. Can I see you guys? Hang on.
Yeah, I'll switch it
here
over
we
so
are.
you can see us now.
So Damian, of course, so one of the big problems is to go from in general, is to go from the peptide to the right protein match.
Right.
And when you go to the protein, now you can have many more multiple sites for phosphorylation. There's also literature about alternative glycosylation versus phosphorylation at some sites.
Right.
Do you have advice or experience about those steps?
We played, I've gotten contacted about Lucifer 2 to try and look at multiple modification events. And I haven't heard back from them, but from what I understand is if the same residue is being modified, you're going to have low scores overall, because the peak shift, for example, with glycosylation versus phosphorylation is very different, right? Glycosylation only shifts the mass by 1 Dalton approximately, and phosphorylation shifts it by a lot more. So when you have a mixture of potential PTMs on a peptide, if they're on the same site, you end up one of the 2 modifications predominates overall, and that's not very helpful in general. But if you have modifications like, say, iTrack and phosphorylation, that the eye track is fixed, so there's no ambiguity there, and that seems to work fine through the program. But if you're trying to find multiple modifications in a peptide or in a search, fortunately there aren't that many that are labile, so you could focus. You can do a search with glycosylation and phosphorylation, and run Lucifer on the phosphorylation site, which is going to be more ambiguous.
Okay, other questions? Well, there's a question about stoichiometry.
Yes.
Maybe I'm out of date, but I believe there's quite a lot of evidence that to switch a protein with kinase phosphorylation in a pathway, you don't have to have 100% occupancy of the site. Sometimes 20 or 30% would be sufficient to achieve the switch. So this means that in your peptide analysis, even though that peak is so high from the neutral loss 98, it may be only a minority of the molecules of the peptide that actually have a phosphorylation. Is that something that you deal with in some way?
Lucifer doesn't deal with that. If you're talking about from a quantitative point of view, if you want to know how often a particular site is modified in a peptide versus another site, I have seen people who will count out, do spectral counting on a particular, Lucifer output and look at how often they have evidence for site A versus site B in the peptide sequence. Although I don't know of anyone who's followed that up with actually looking at the chemistry of the stoichiometry there. I'm not sure if that answers your question or not.
Well, I think it might be important. Maybe it's not. I just don't know.
That's a hard one to. That's a biochemist problem, which
right.
Much harder than my problems.
Okay. Well, we had a really nice workshop back on the 2nd and 3rd of March. All of our proteomics informatics students and Lexi, of course. But I don't think anybody is here from that group today, so they're lost. And I think this is tremendous what you're doing. You have any other comments you want to give us?
No, I mean, thank you for arranging this. I mean, I know Alex probably had to go to a lot of trouble to have me speak from this far away.
Well, it saved you a trip. That's a lot of hours. It's good. Although
yeah, tell me about it.
See you anytime. Any other comments?
Thank you again.
Alex, you were part of the credits on the whole design of the project.
Not exactly.
Credits are he got credit for getting this off the ground because, the IT part of this is the hardest part. Getting it to be able to present to you guys is probably the hard part in my opinion. Because anyone who's trying to make a connection across Skype will tell you it never works out the way you think it's going to work.
Okay, listen, our best to you and your colleagues there, and thank you for connecting with us and giving this up-to-date presentation. Very good.
Thank you.
Thanks.
That wasn't a real question.